The topic was the fact that we are increasingly working on studying how tumors respond to drugs using the genetic of the tumor. And the foundation for that has been this theory of oncogene addiction. That is, if you have a mutation in an oncogene, then shutting down that oncogene will actually cause the tumor to die. And unfortunately, this has really failed a little bit to deliver on promises. In some cases, it worked exceptionally well, but in a very small number of patients. And so I was reflecting on what are the reasons why this has failed and what can we do to address that. And in particular, what causes tumor initiation and progression is actually not the individual mutation. It's the specific pattern of aberrant activity of the proteins, relatively small number of proteins. And you can actually activate a protein by mutating it, but that's just one way to do it. You can do it in quadrillion, quintillions of other ways, by mutating upstream regulators, interactors, all sorts of other things. And so what we decided to do is that instead of just measuring the mutation in the proteins or in a corresponding gene, we should perhaps think of having better methods to understand whether proteins are aberrantly activated. And we do that using the targets of the protein as the best reporter. Basically, if a protein regulates some targets, if those targets are differentially expressed, then this protein is active. If the targets are not differentially expressed, then it's not active. And that has turned out to be a phenomenal predictor of drug activity and response, clinical response to patients. And has also generated a large number of new discoveries that we've published over the years, including the ability to identify proteins that don't work individually but in pairs, so that one or the other doesn't do anything, but the combination really induces very strong tumorigenesis, and silencing both proteins induces remission. So this is, for instance, a clinical trial that we have just initiated in breast cancer. These are patients that have HER2-positive breast cancer that have failed therapy with trastuzumab, which is the inhibitor for HER2. And what we discovered using this approach, by studying all the regulator proteins that are not mutated in the cell that has HER2 amplifications, we discovered that a particular protein called STAT3 was actually aberrantly activated but never mutated. And downstream of these proteins, there is secretion of a protein called interleukin-6 that goes outside of the cell, binds the receptor, and activates a pathway called the JAK-STAT cascade, which ends up on STAT3. So you start a loop that is a really vicious loop, because now you can turn off HER2 and the loop keeps going. So by using another drug that is an inhibitor for the JAK kinase, which is called ruxolitinib, we were able to induce very strong synergistic response in patients. And now 4 patients have been enrolled in the trial. 2 have actually responded, and the one died for a completely independent reason. And one that has not responded was actually predicted not to respond because he had ER-positive state, and we predicted that this would only work in ER-negative state. So it is a very interesting way of doing it, and there are many other examples. That's a different trial. The 260 patient is a really unique trial, which we call an N of 1 trial, where every patient is actually treated with an independent strategy, because we're using that trial to actually understand patient response. And so for every patient that is enrolled in that study, which covers 9 rare or untreatable malignancies, we are generating a complete map of the protein or proteins that the patient tumors is dependent on. And then we figure out how to match them to very specific drug or drug combinations that can be tested in a mouse where the patient tumor has been transplanted. And then we can advise the physician, the treating physician, about the drug or drug combination that worked. So some of the really interesting research that has now profound clinical implication is in the identification of biomarkers that can predict, for instance, whether patients with a prostate cancer will actually die or the disease or not. For instance, in prostate cancer, over-treatment is one of the major problems. And in fact, most prostate cancer patients receive either therapy or most likely surgery that they may not have needed. And so what we did in that case, this is a collaboration with Corey Abate-Shen and Michael Shen at Columbia. We showed that by using this approach, that is by looking at the target of the protein, we were able to discover 2 proteins, one called FOXM1 and one called CNPF, that in isolation do absolutely nothing. But when they're co-active in a tumor, they are just the harbinger of the worst possible response, because they actually regulate the entire set of programs that are associated with metastatic progression, etc. So if you shut down one or the other, nothing happens in this tumor. You shut down both of them, this tumor dies. And more importantly, at diagnosis, that even in a cohort of 900 patients that were followed up for 20 years, you can see that the patient that had the double negative, that is, had no activity of either protein by, it's called immunohistochemistry, where essentially none of them died. Literally about maybe 3 patients died. And in the cohort of the double positive, which had basically both protein active at diagnosis, that represented 90% of the burden of the tumor, with an extra 10% represented by the single positive for one of them called FOXM1. So this basically tells you that, and in fact now it's been developed into a clinical biomarker, it has been licensed, and the idea is that these essentially represent one way in which at diagnosis you can predict with far window of opportunity of many years whether the patients will have a very aggressive type of tumor or not. The foundation of this entire approach that I told you is that we can actually measure the activity of the protein by measuring the expression of its targets. Okay? It's like saying that I can figure out who is the person that committed a crime but is sitting in the shadow by knowing who was at the crime scene and knowing who are the friends of that person. Now, the big problem that people have not yet been able to solve is to figure out in each cancer context what are the targets of each protein. It's called systems biology because it means that you have to build a regulatory networks that has millions of interaction, where every regulator protein out of 6,000 proteins that are either transcriptional regulators or signal transduction regulators is associated with somewhere between 50 and 1,000 targets. And so that analysis is done with algorithms that have now been extremely experimentally validated in the lab, and that typically have 70 to 80% accuracy in predicting true physical targets of the proteins. And only when you have these targets resolved in each tissue type, each tissue corresponding to a tumor, you can now apply this approach. Without that, all the different assays that we have to measure protein activity are not really very good, because the measuring the RNA is like many sort of step removed from the protein activity. Even measuring protein phosphorylation is very noisy and actually has very large variability even within a sample of a patient, because if you leave a sample out for 5 minutes versus 10 minutes, the phosphorylation state on the protein will change. So the actual expression of the targets is one of the most stable and most reproducible predictors of the aberrant activity of proteins. So what we do is the following. First of all, we build a model. We have to build this model of about 1 million regulatory interaction. For that, we use a large cohort of patients, typically 200, 300, 1,000 patients, as has been made available, say, by TCGA. We now don't need to do that anymore because we've built so many of these models that we can actually take a tumor that has never been, that is very rare, for instance, and there's not enough patients, and we can use the combined knowledge in all of these models to predict what the targets are. Then we take the sample of that particular patient and we run it through the model and we ask, well, if this is the differential expression of the genes in this particular patient, and these are all the interaction, then what are the proteins that are inducing that differential expression? And that gives us a very short list, typically about 20 activated and 20 inactivated proteins that we call the master regulators. We can now match those to the response of drugs. So we take a large repertoire of FDA-approved and developmental drugs, and we ask the opposite question. We ask which drug takes the specific targets of the protein that we want to shut down and completely flips their status. So if they were overexpressed, now they're underexpressed, they were underexpressed, now they're overexpressed, because those drugs will actually inhibit the activity of that protein. And so we can match a drug to the activity of all of these proteins. We can match drug combination to the activity of all of these proteins. And what is important is that when you actually hit the right targets, the entire master regulator signature collapses. So you have an effect where you don't have to target a single protein, but you're actually targeting an entire module of proteins. They all work together to support the activity of the tumor. And so this is actually shown that you can, with pinpoint precision, identify drugs by studying only the human tumor that you can then put in a mouse xenograft that is in a patient transplant to an immunocompromised mouse, and will actually put the mouse in remission without ever having been screened in cell lines or in mouse models. Well, the take-home message is that we have to abandon what is easy to do, and right now, sort of discovering mutation is very easy to do because sequencing technology have become extremely cheap, and really buy more into more integrative approaches that bring both the mutational status, the status of protein, the status of RNA, etc., and use that as a compendium of information to give us a full perspective on the vulnerabilities of tumors. And this actually doesn't just work in tumors. We've just published the same exact methodology in Parkinson's disease, in ALS, in Alzheimer's, in alcohol addiction, and in stem cell pluripotency. So this is a very general paradigm. It's not just restricted to cancer biology. So if you use these integrative approaches, you tend to be able to extract more information, but some of them, unfortunately, are complex. They're not easy, and that's why we have to use a systems approach to bring this to the patient.